In this video, we will discuss about limited proteolysis coupled mass spectrometry. This is an advanced form of mass spectrometry in which we can use these techniques to detect whether there is a structural change in the protein or not. In the traditional mass spectrometry, we can only detect the abundance changes in the protein. However, by using this workflow, we can detect we can also detect whether there is structural changes in the proteins or not. So let us go through the workflow in brief. So in this workflow, we have to double digest our samples along with the control samples. So the first step is to take proteins in its native condition or extract proteins from in its native condition and digest it with some proteases that has broad specificity. As in the one condition one, or we can say one condition is of the wild samples that are collected from the wild type, and in another conditions samples that are collected in our interested form, or like some kind of gene knockout form. So and the arrow shows the digestion site for uh, the proteases that has broad specificity. So in this, the first digestion has been done in its native structure by some broad by some protease that has broad specificity. And after that, what will be done is, after that, the a regular process, that is denaturation and alkylation, will be done of the samples, and then the uh, tryptic trypsin digestion specific proteases that cleave of the protein at specific site, that proteases will be used, such as lyse or trypsin protease will be used to digest the proteins afterwards. This is the second digestion of the samples, and after that, the samples will be run in the mass spectrometry. And in the mass spectrometry, after compensating for the relative abundance of the protein uh, with the help of abundance of proteins in the wild type, then the structure then the structural changes of the proteins can be identified from the by comparing the abundance of the peptides that are complete trypsinic peptide and half trypsinic peptide, or incomplete trypsinic peptide. So what does this mean? This means that suppose this side that is shown in the yellow color is cleaved off in between, whereas there is no yellow color sides. So this amino acid sequence will not be cleaved off. However, it will be cleaved off by trypsin. So when this is cleaved off by trypsin, this forms a single peptide, whereas when this thing is cleaved off by trypsin, this forms a double peptide. So when this will run through this peptide will run through the mass spec, what will happen is that in the regular or control sample, we will see a peak for this only one peptide at a higher site, whereas when it runs for a conditioned sample or sample of interest, then we will see different peaks of this half tryptic half tryptic peptide, and we will see a low peak or no peak of this complete trypsin peptide. And so this is one of the example by which we can use this limited proteolysis coupled mass spectrometry to detect the structural changes in the proteins. And for this, we can use different kind of proteases. And if you need to understand the effect of different kind of proteases in the limited proteolysis coupled mass spectrometry, you can go through the reference that has been provided in the description. I hope you have understood something about this limited proteolysis coupled mass spectrometry and its workflow, and how it is able to detect the structural changes in the proteins. If you need to understand this thing in details, go through the reference that has been provided in the description. That's all for this video. Thank you for your time.